Today, I am happy to be able to present the work I've done in the Joe Wong lab to develop this mouse model to determine the functional impact of prostatic E-cadherin loss on the prostate and bladder. With this talk, I'd like to also highlight how CAIRIBU has influenced the development and validation of this model and was really critical for helping us to move this project from an idea to a reality. In thinking about how CAIRIBU has influenced and fostered collaboration in the development of this mouse model of E-cadherin loss in the prostate, I wanted to touch on four main types or ways I feel CAIRIBU has enhanced this project. 1, helping with idea development. 2, experienced advice and insights from the CAIRIBU community. 3, funding and sharing of resources. And 4, fostering discussions from researchers doing related studies. In 2014, Katie Willett, a technician in the Wong lab, identified PSA protein in the stromal compartment of BPH tissues by proteomics analysis of laser capture microdissected stromal cells. Immunostaining and ELISA analyses confirmed the presence of PSA protein in the stromal compartment, and in situ hybridization confirmed that PSA mRNA is expressed in prostate epithelial cells, confirming that the PSA observed in the stromal compartment was not generated by BPH stromal cells. Joe discussed these findings with Will Rickey, who confirmed these observations of PSA protein in BPH stroma at the University of Wisconsin. These findings suggested the possibility of alterations or dysfunction of the epithelial barrier in the prostate, possibly resulting in the leakage of secreted proteins such as PSA into the stroma. This could cause an inflammatory response and might play a role in BPH pathogenesis. We received U54 O'Brien Center funding from the NIDDK in 2016 to study the potential role of epithelial barrier alterations in BPH pathogenesis. As part of the U54, we held an annual meeting where Janaki Gustafsson, who is pictured on the left, was an invited keynote speaker because of his work showing E-cadherin was decreased in BPH tissues. In the audience that year listening to Dr. Gustafsson's talk was Li Feng, identified by the red circle in the lower photograph. Leaf confirmed this decrease in tissue specimens from BPH patients and showed that E-cadherin loss resulted in an increase in epithelial permeability in prostate epithelial cells using cell lines that we got from Simon Hayward. Since conventional E-cadherin is expressed by both luminal and basal epithelial cells in the prostate and is critical for normal prostatic development, we utilize the luminal-specific PSA Cree ERT2 mouse developed by Drs. Pierre Chambon and Daniel Metzger. We had previously used this mouse to lineage trace how luminal epithelial cells are regenerated in the murine prostate. Mice with prostate luminal epithelial specific deletion of E-cadherin were generated by crossing the tamoxifen-inducible PSA Cree mouse with the Cdh1 floxed mouse from Jackson Labs to generate mice hemizygous for the PSA Cree and heterozygous or homozygous for the E-cadherin floxed allele. Tamoxifen administration was performed at 7 weeks of age when male mice reached sexual maturity. In discussions of our planned studies at Asilomar, Mark Nelson, pictured on the right, suggested that tamoxifen has been reported to have an impact on prostate, and to control for those effects, all cohorts, including the controls, should be treated with tamoxifen. More recently, at the virtual CAIRIBU meeting last year, Dale Bjorling suggested we confirm that there was no prostatic phenotype in the PSA Cree mouse, and that there was no PSA Cree activity in other organs in our mice. We have incorporated both of these excellent suggestions in our experiments, and we looked at these animals at age 6 months and 24 months. Homozygous deletion of E-cadherin in the murine prostate luminal epithelial cell induced epithelial proliferation, inflammation, and increased fibrosis in the prostate. Chad Vizina has provided guidance for conducting void spot assays as a CAIRIBU and GoodMap resource, and we found via void spot assays that homozygous mice had an increased number of bladder voids and smaller voided volumes than their age-matched controls. Cystometry showed that E-cadherin knockout mice also developed an overactive bladder phenotype by 6 months of age, similar to other murine models of prostate inflammation. Mice with heterozygous deletion of E-cadherin did not display any changes in the prostate or bladder at 6 months of age. And we recently published this work in Endocrinology in January. Although homozygous E-cadherin deletion induced prostatic inflammation and bladder overactivity, which are associated with human BPH and LUTS, there are some major differences between these observed changes in mice and what is seen in BPH patients. For one, these changes were observed at 6 months of age in mice, which correlates to around 30 years of age in humans, well before BPH LUTS develops in men. Additionally, loss of E-cadherin resulted in the persistence of E-cadherin negative cells surrounded by E-cadherin positive cells derived from basal progenitors. This is not what we observe in human BPH, where E-cadherin staining is not seen in the same mosaic pattern. To see whether the heterozygous model was more similar to human BPH and LUTS, we examined these mice at 24 months of age to see if aging combined with partial E-cadherin deletion would impact the prostate or bladder. Similar to BPH and LUTS, which typically develop after 50 years of age in men and affects almost 80% of men by the age of 80, heterozygous mice did not differ from age-matched controls at 6 months of age, which, as I mentioned previously, is around 30 years of age for humans, but did develop prostate inflammation and bladder overactivity at 24 months of age, which is around 80 years of age for men. Interestingly, the prostate epithelial cells in heterozygous mice displayed areas with apparent decreased immunostaining of E-cadherin similar to the staining pattern observed in human BPH epithelial cells, as well as the mosaic pattern of E-cadherin negative and positive cells observed in the homozygous knockout mice at 6 months of age. Teresa Liu is a member of the CAIRIBU group who is very interested in the role of aging in BPH and LUTS development. She's recently published a paper showing that as mice age, they develop prostatic defects and changes in urinary function that are associated with BPH and LUTS. These changes include increased fibrosis, prostate enlargement, as well as smaller and more frequent bladder voids. So we wondered if similar changes occurred in our animals. In a post-hoc comparison of our control mice at 6 months of age and at 24 months of age, we found that there was an increase in prostate inflammation and a significant decrease in the bladder average voided volume. Older mice also had a tendency to have an increase in the number of non-voiding contractions, increased urinary frequency, and a decrease in total urine. These findings are similar to those observed by Teresa Liu in her study and suggest that aging contributes to the development of BPH and LUTS characteristics. These findings suggest a possible synergism with E-cadherin downregulation in BPH. We also found that E-cadherin immunostaining was negatively correlated with aging and prostatic inflammation in human prostate tissues. These findings suggest that the aging prostate is characterized by an increase in inflammation and a decrease in E-cadherin. The mouse model suggests that decreases in E-cadherin also can contribute to prostate inflammation, and that this inflammation is accompanied by an increase in bladder overactivity, suggesting that aging combined with E-cadherin loss could further increase inflammation and possibly contribute to BPH and LUTS pathogenesis and symptomatic progression. Other ideas and insights have come from conversations I've had either with CAIRIBU researchers or from researchers at CAIRIBU-sponsored meetings. I pictured some, but not all of you here. Although targeting E-cadherin directly presents a daunting challenge, targeting prostate inflammation to reduce or improve BPH symptoms could be much more feasible and could also be explored with this mouse model. We don't fully understand the mechanisms of aging and how it impacts E-cadherin and BPH and LUTS pathogenesis yet, but there are possible lifestyle changes being explored that might hold promise for targeting inflammation and slowing the aging process. Thank you all very much for your attention.